clusters of adaptive alleles in globally distributed species.
Okay, hi everyone.
Oh, sorry. Wait for the bell.
Oh, the bell!
Wait for the bell.
Okay. Hey everyone, my name is Kieran Samuk. I'm a PhD student at the University of British Columbia in Dolph Schluter's lab. And I'm going to talk to you today about a project that I've been working on with 5 others at UBC, looking at how gene flow can shape the genetic architecture of adaptation. Okay, so as I said, this was a collaborative project between myself and 5 others, and here they are. Greg Owens, Anna Redison, Sarah Miller, Kieran Delmore, and my advisor, Dolph Schluter. So if you don't like anything you hear today, these are the people who need to be blamed. Okay, so hundreds of studies now have found signatures of selection in the genome. So here are 3 reviews sort of summing up some of those results. So it goes without saying that we know that these signatures of selection are ubiquitous in the genome. They're kind of almost everywhere that we look. And so here's an example of some of these signatures. So this is from the Heliconius literature. And this is a trace of FST, which is a measure of genetic differentiation between 2 different pairs of Heliconius populations. So one of the striking things about this is if we look at FST across the genome in this top case, we can see it's highly localized in a couple places in the genome, whereas it kind of seems to be lousy with divergent regions down here. So why do we see adaptation in some regions of the genome and not others? What's causing this? And what I'm interested in is, are there general constraints on where adaptation can occur in the genome? Or can it just kind of happen anywhere that adaptive variation happens to be available? And so what I'm going to talk to you about today is some recent theory that suggests that gene flow, migration between populations, can actually constrain where adaptation occurs in the genome. And I'll be kind of loosely referencing the predictions of these 2 models, one by Berger and Ackerman, and another by Yeaman and Whitlock, both from 2011. So I'll just kind of quickly go through the basic idea of these models so you can get a sense of where these predictions come from. So the scenario is we have divergent selection. A single population splits in 2, colonizing 2 different environments, and initiating divergent selection. If we look within each one of these environments, basically what's happening is selection is building up sets of selected alleles. So different mutations occur on different genetic backgrounds and are kind of stuck together into one background by selection. Selection is building up these associations. So what happens when we add gene flow into the mix? Well, if we imagine our migrant haplotype invading our blue population here, if it interbreeds with a local type, it's going to start breaking apart those associations that selection has worked so hard to build. So there's basically a tension between selection building up these sets and gene flow and recombination breaking them down. So kind of the key result from these models is that tight linkage between adaptive alleles can actually prevent these sets from being broken apart. So if we imagine our migration scenario again, where we have actually variable architectures of fitness. So we have, for example, one case, a loosely linked haplotype, where the adaptive alleles are far apart of the chromosome, compared to a tightly linked haplotype, where they're packed together all nicely. If we look at what happens with hybridization and recombination, you can see that in the top case, that association has been broken. These are now on different backgrounds. Whereas in the bottom case, that association is maintained because of that tight linkage. So if we consider, for example, this to be a generation, this guy here is going to have the highest fitness because it's managed to maintain that LD. Okay, so this is really sort of that prediction in words. Divergent selection of gene flow favors the evolution of tightly linked clusters of locally adaptive alleles. So what we wanted to do was actually test this prediction empirically. And to do that, first I'm going to refer to this divergent selection of gene flow as DSGF, so I don't have to keep, it's barely fewer syllables, but one problem with testing this idea is that all kinds of other processes can cause cluster signatures of selection of the genome. So one of those, very basic one, is that genes are not randomly distributed on chromosomes. So for example, if we look at gene density, that alone can cause non-random clustering of adaptive alleles. Same goes with mutation rate. We have to control for those. A more insidious problem is that selection itself can cause cluster signatures. And for example, in the case of a selected sweep, where we have a beneficial mutation pulling all these other alleles to fixation with it, that generates a clustered signature of selection of the genome. We rely on that fact when we do tests of selection sometimes. We need to deal with that. So luckily there are solutions to both of these problems. in the case of gene density and mutation rate, we can statistically control for them. We can estimate these things. These can serve as basically the null hypothesis. In the case of hitchhiking, what we can do is we can compare our DSGF to divergent selection alone and gene flow alone to get a sense of whether or not there's something special going on in this case. So just to kind of outline how that approach works, basically our idea was to classify pairs of populations based on the presence of selection and gene flow, and/or gene flow. Then we wanted to find loci that are adaptively differentiated between them. Finally, we wanted to quantify the degree of clustering. So this is really that formal test of those theoretical predictions while controlling for mutation and gene density. Okay, so perhaps you saw it coming with the Dolf-Schluter. Sticklebacks are an excellent system for applying this approach. And there's two main reasons for that. One is that we have really solid knowledge with the genome of the stickleback. So we know lots of stuff about things like recombination rates. We also have a high quality genome, and so we can estimate things like gene density, and using different kinds of methods, also mutation rate. So that gives us that statistical control. Most importantly, however, for the test of this particular hypothesis, sticklebacks have repeatedly colonized all different kinds of environments, all over the northern hemisphere, and adapted to them in the presence and absence of gene flow. So that really gives us that raw variation we need to be testing this theoretical prediction. Okay, so basically what we did, we went to all these databases and downloaded all the stickleback data we could get our hands on. It's kind of a moving target because there's so much stuff being published all the time. But we ended up with a huge amount of data from many different pairs of populations in the world, many thousands of individuals, different sequencing technologies, et cetera. And we used a common bioinformatic pipeline to identify variants of these guys. So back to our approach here, now that we have these populations in hand, we want to classify them so we can compare them based on the presence and absence of selection of gene flow. So here's kind of the idea. We want to do these statistical contrasts between these different regimes. So we have selection here, diversion and parallel selection, gene flow and allopatry. And basically the idea is, here's our DSGF comparison here. And if we just map those theoretical predictions on, we're expecting the most clustering of alleles, packing those alleles together. In this case, you might expect a little bit here because of hitchhiking, for example. That's just divergent selection alone. And we don't really expect to see anything going on here. Theory doesn't really make any predictions about those. Okay, so just to remind you, this is our key prediction here. And so to get at selection, we use environmental differences as a proxy for divergent selection. So this is probably pretty reasonable for sticklebacks, but so for example, we consider these benthic magnetic types to be divergent, whereas we consider two different marine populations from different locations parallel. So that's probably a pretty reasonable assumption for sticklebacks. In the case of gene flow, we use geographic distance and the presence of physical barriers as a proxy. So basically, all the pairs of stickleback populations that people have studied, in which there's been evidence of recent gene flow, share these two characteristics. No physical barriers within 500 kilometers of one another. So that's a very basic proxy for gene flow. Okay, so now that we had those populations in hand, we wanted to find loci that are actually adaptively differentiated between each pair. And so we took a basic approach. We used an FST outlier approach. So FST is a relative measure of allele frequency difference. Basically, the idea is loci with extreme values of FST are going to be enriched for things involved in adaptation. They won't all be adaptive loci, but they should be enriched for adaptive loci. Okay, so we carried out many FST genome scans, sometimes many genome scans in the case of divergent selection allopatry. But just to give you a sense of the sample sizes for each one of these regimes. So here's some example data. So this is FST plotted on one chromosome and one pair of populations per regime. And so you can see, for example, in the case of parallel selection of gene flow, FST is low. We expect that. Gene flow with parallel selection should have low differentiation in general. If you look at the allopatry case, it's kind of like this crazy solid block. In the divergent selection case, however, we're seeing very low FST at the periphery of the chromosome, where it gets higher in the middle. If we apply a smooth to that, see we're getting kind of these flat lines and these different, all these other regimes, and we're getting this crazy hump shape up here. So I'll come back to that. So that's just an example of what the raw data looks like. Okay, so we have our populations. We know the loci that are adaptively differentiated between them. Next, to test this theory, we really want to quantify the degree of clustering of these loci. So just to remind you of theoretical prediction, we should see tightly linked clusters of locally adaptive alleles in these TSGF populations. So to actually get at this, we took a very basic approach. I'll spare you the details, but we basically generated a null expectation based on sampling nearest neighbor distances between all the SNPs in the genome, and then compared the actual empirical distance between the outliers to that expectation. And so we basically generated a score for outlier clustering. So here are the results of that. So on the y-axis is this outlier clustering score, so a higher value on this y-axis means those outliers are more packed together in the genome, more tightly clustered in terms of genetic distance. So basically what you can see is outliers from DSGF comparisons were unusually close together in the genome. So just to remind you, there's a DSGF population, gene flow alone, for selection alone either. And we assess the significance of this using a permutation test. Oh yeah, so each one of these points is actually a different comparison, just to give you a sense of how much data we ended up having. So that's great. As predicted, adaptive alleles are more tightly linked in these DSGF comparisons. So we wanted to probe that a little bit more and ask, what does this actually look like in the genome? Like, what's going on here? And so here's back to our FST trace. And so what's kind of interesting is that if you map on recombination rate, so this is just a smooth recombination rate, most stickleback chromosomes that are metacentric, that have the centromere in the middle, have this U shape for recombination. And so what you can see is that in the divergent selection of the gene flow case, there's this clustering of adaptive alleles in regions of low recombination. So that's how that's actually being achieved. So as predicted, DSGF favors tightly linked clusters, and this seems to be being achieved by a concentration of these alleles in regions of low recombination. Okay, so the theoretical prediction of these two papers were upheld. So I'll quickly go through the implications of this. So kind of what we've been thinking about a little bit is that gene flow may actually make the usable area of the genome effectively smaller. So based on our data, when we look at allopatry, it seems you can kind of diverge wherever you want here. But it doesn't matter with the recombination rate, whereas once you add gene flow into the mix, it's kind of limiting the available substrate for adaptation. Smaller set of genes basically can't diverge outside of these regions of low recombination. So what does this mean? If we have a smaller set of genes available to adaptation, one kind of odd outcome of this is that we might actually see more reuse of the same genes because there's a smaller set to draw from. Another strange implication of this is that by reducing the substrate available to adaptation, gene flow might actually be making less of variation overall available to adaptation. I think this might be a novel effect of gene flow. Okay, so just to wrap up, gene flow favors the clustering of adaptive alleles, regions of low recombination, and this may be an important general constraint on adaptive evolution. And I have lots of people to thank, and especially the organizers of the Symposium. Thank you very much. I think I have time for questions. Yep.
So in addition to clustering by physical distance, it seems like the same logic would also predict clustering on the same chromosome.
Yep.
Did you test for that, and did it turn out so smashingly?
We did, and it did. I don't have a plot to show it, but we basically also came up with a score of like localization, I guess genome-wide clustering versus chromosome-wide clustering, and the results are similar. Yeah.
So I'm imagining the situation where you've got conflict between the availability of sharing alleles between universally adaptive alleles and being able to maintain locally adaptive. So in theory, when should that start to break down? When should that conflict come to hit a brick wall and lead to speciation?
So the idea is like there are things that are universally adaptive that you want to let through. Is that what you're suggesting?
Yeah. So at some point there's going to be conflict that's going to be too much, right? The benefit of those universally adaptive alleles is going to be insufficient to work its way in. So I mean, in theory, has that been worked out? We have an idea of how complex the genomic architecture of these traits can be.
Yeah, I'm not sure. I know that Sam Gaiman is working on similar stuff to that. My guess would be that it would have to do with the strength of divergent selection between the two populations. But I'm not totally sure. Thanks very much.